we're going to do a high-speed run through cell respiration. So what we're looking at here is a mitochondria, which is the organelle where cell respiration occurs inside of eukaryotic organisms. Now you don't need mitochondria to do cell respiration. Bacteria and other prokaryotic organisms have been getting along just fine without them. However, they're way less efficient, which is why, part of the reason at least, why they're so small. So the magic of the mitochondria is all of these many folds. These various folds on the inside create a ton of surface area, and across surface area you can do a whole lot of different reactions across those membranes. What's kind of cool about this diagram, even though it's from 1998, is that it shows our ATP synthase here. So this guy, these little brown doohickeys, are ATP synthase proteins, and you can see how they're just all over that inner membrane of the mitochondria. And that's going to be pretty important when we get to the electron transport chain. Looking at the whole process is crazy. However, you break it down piece by piece, I think it makes a lot of sense. We're sort of looking at photosynthesis in reverse. We're taking glucose and we're breaking it down and we're releasing the energy that has been stored up inside of it. Now that begins with glycolysis. So we'll get rid of the rest. With glycolysis, what's happening is we take our glucose, and the first thing we need to do is we need to spend 2 ATP. So we got minus 2 ATP. Right away we've lost that ATP. That goes into changing around the molecule of glucose and preparing it to be broken down. During this process it becomes an unstable molecule and it splits into 2 3-carbon molecules. That's our G3P, which actually we saw in photosynthesis. Now those 2 molecules then are both going to go through the remainder of this process, and what that's going to net us is 2 NADH are going to be reduced from NAD, and that is going to be one of our major energy carriers in this process, and we're also going to make a total of 4 ATP. So our net gain here, if you get rid of that minus 2, our net gain here is going to be 2 NADH and 2 ATP, and that's for glycolysis. The other thing that's going to be coming out of glycolysis is 2 molecules of pyruvate. Now the pyruvate itself is going to have to be changed around a little bit before it can be moving into the Krebs cycle, but what you see here is that the pyruvate is actually going to pass through the membrane of the mitochondria into the matrix on the inside. And the matrix of the mitochondria is just the fluid portion here. During that process it's going to change around its own form. It's going to become acetyl-CoA, and then eventually it's going to be a 2-carbon molecule that's coming in here to the Krebs cycle. So let me show that here. So we have a 2-carbon molecule coming in. Now, it was a 3-carbon molecule, so what that means is we actually lost a CO2, and we also generated in that process an NADH. So I just want to add those to this diagram. Continuing on with the cycle part of this, oxaloacetate is a 4-carbon molecule right here, and the significance of that is that it's going to scoop up the 2 carbons from the pyruvate, or what was once the pyruvate, and it's going to become a 6-carbon molecule called citrate. You don't need to memorize the names of all these molecules. That's not really the focus of what this is about, but understanding the general process and why it is that these things need to happen this way and changing form and getting these molecules to the right state in order to maximize the energy we can pull out of them, that's pretty important. Continuing this process, so right now we have we've made 1 NADH, and we've also lost 1 CO2. Coming in here we have again capturing some energy and producing another NADH, and we lose another CO2 as waste. We're going to breathe that out, which is kind of weird. It was once part of like your sandwich and now you're breathing it out. We make another NADH and we lose another CO2. Okay, and we're also going to make a different energy carrying molecule called FADH2, and we're going to make, just for good measure, we're going to make 1 more NADH over here. And now we're back to the beginning of our cycle. We scoop up another pyruvate and we do it again. Okay, so if we're tallying this up here, we are going to get a total of 4 NADH, 1 FADH2, and I just realized I missed right here in the middle. We also generate 1 ATP. So the total gain from the Krebs cycle and from each pyruvate that goes in here is 4 NADH, 1 FADH2, and 1 ATP. So the ATP obviously is our end goal of cell respiration, so that's good. We got one of those. But we right now are mostly focused on the NADH and the FADH2, which are energy-carrying molecules, but not nearly as useful kind of cell-wide as ATP. So we've got to get those changed into ATP. And that is where the electron transport chain comes in. So jumping back to our full picture here, we see the electron transport chain actually happens right here inside the inner membrane of the mitochondria. So the NADH and the FADH2 that are being produced inside of the Krebs cycle and also out here in glycolysis are going to make their way to this inner membrane where they're going to donate their electrons. The way that happens is the NADH is going to be oxidized, it's going to lose its electron, it's going to split off its hydrogen ion, which is going to be a positive hydrogen ion or just a proton. The electron is going to go through a pathway of proteins on the membrane here, and that's going to donate its energy to allow for active transport of these protons out of the mitochondria's matrix and into the inner membranous space here, and that's going to generate a really big concentration of hydrogen ions over here. Meanwhile, the FADH2 is also donating its electrons for the same process. Those electrons are going into pumping even more hydrogen ions. The hydrogen ions, again, are coming from the FADH2 and the NADH2 that as they're oxidized, they lose their hydrogens. Okay. Now, important here is to understand what the heck the point is of moving hydrogen ions. That alone hasn't really accomplished anything. But if you think back to photosynthesis with photosystem 2, and when it was doing the exact same thing, the generation of a hydrogen ion gradient is going to push these hydrogen ions, or it's going to encourage them rather, to get back across that membrane. And that's where ATP synthase, those little brown knob things we saw in the mitochondria picture, that's where those come into play. They offer a place for those hydrogen ions to flow back through. However, the movement of those hydrogen ions changes the shape of this protein and allows it to capture some of the energy of their movement, and that's going to allow it to generate ATP. In the end, each NADH, which we actually had about 10 NADH total. Doing some bookkeeping, we had for each pyruvate that we had, we generated 1 NADH in glycolysis and then 4 more in the Krebs cycle. So there are 2 pyruvates, so we ended up with 10 total NADHs. And since each pyruvate generated an FADH2, we therefore have 2 of those as well. Each NADH on average, or actually I think maximally, can generate about 3 ATP. And each FADH can generate about 2 ATP. And that's just how much ATP they can generate from the movement of hydrogen ions here. It's not a one-to-one of hydrogen ions into the cell to ATP, but the math works out to about this. So we've got about 30 ATP being generated by the NADH and about 4 ATP from the FADH2. Now, reminding ourselves that in glycolysis we had 2 ATP, and in the Krebs cycle we also generated 2 ATP, 1 per pyruvate. Then that gives us a grand total of 38 ATP. Now again, that's kind of a maximum number. It's usually anywhere between like 32 and 38. That's just because of the chemistry of what's going on inside of the cell and inside of the mitochondria. An important thing that we missed or just haven't talked about yet is what the heck happens to these electrons. So they go moving through these proteins and they're donating their energy and they're losing that energy, and now they just come right back into the matrix. Well, they have to be scooped up by something, otherwise we're going to get this abundance of electrons, and that's going to cause all sorts of problems. So this is why we actually need to breathe. If you've never stopped and wondered why it is you breathe, you should. And this is the reason. So the oxygen comes in and actually is going to capture these excess electrons. It's going to use those electrons along with 2 hydrogen ions or protons to produce H2O. And this is why when we talk about cell respiration, one of the products of that cell respiration is water. The water is generated by the oxygen here gaining electrons, being an electron recipient so that we don't have this overabundance of electrons. So if we recap this, going back to the full diagram here, we have glycolysis happening in the cytoplasm where we have glucose being broken down. It costs us 2 ATP, but it gets us 4 more, so that gives us a grand total of 2 ATP, as well as a couple NADHs. We then move our pyruvate into the mitochondria where the Krebs cycle occurs. It gives us a whole bunch more of these NADHs. It gives us FADH2 and some ATP as well. Those electron carriers, the NADH and the FADH2, then go to the inner membrane where they go through the electron transport chain. They donate their electrons, they pump hydrogen ions, and that buildup of a hydrogen ion gradient is going to then facilitate ATP synthase, making ATP, tons of it, and in the end those electrons will get scooped up by oxygen, and that'll also take care of some of these hydrogen ions and gives us water as a wasted byproduct. So the two byproducts of this process are water and CO2. And that is cell respiration.